Oh my genade.
Ik ga naar de laatste slide. Ik moet daarbij nummer 42 kom ik wel mollen en dan moet dat nou zien. Dus ik weet niet hoe dat nou gebeurt.
Als ik hier ga, dan ik just niet.
Welcome to the inaugural lecture of Prof. Bertie Fielding. Can I please ask you to stand as we welcome the procession inside the venue? Thank you so much. You may be seated. Prof. Moyo.
Okay, good evening, everyone. Okay, we'll try that again. Good evening, everyone, and welcome. That's much better. Thank you so much. My name is Sibusiso Moyo. I'm the DVC for Research, Innovation, and Postgraduate Studies at Stellenbosch University. So I just want to welcome you to this 2025 inaugural lecture by Professor Bertie Fielding, the Dean of the Faculty of Science at Stellenbosch University. So good evening once again. I want to just acknowledge, of course, Professor Bertie Fielding's family, Portia Fielding, his wife. You can just raise your hand so people see who you are. And then their sons, Michael and Daniel, who are here with us. And then Ezra, his son, is busy with studies in France, and he's joining us online as well. And then I also want to welcome his father-in-law, Anthony Scott, who's also here. Thank you so much for joining us, sir. And then also University of Western Cape leadership, Mr. Mani Regal, the ex-director of finance at University of Western Cape, is here. Thank you so much for joining us, so in absentia. And then, of course, friends, collaborators, students, colleagues from the University of the Western Cape and other higher education institutions that are present in South Africa and abroad as well, and those who are joining us online. And from Stellenbosch University, I want to welcome the academic procession, which is led by our Rector and Vice-Chancellor, Professor Diresh Ramjugenath, who's with us here today. And then also Professor Richard Stevens, the Acting DVC Learning and Teaching, who's here with us today. Maybe if you raise your hand so people know, like, I'm Richard Stevens, you know. Thank you so much. And then we've got Professor Assam Sethla, who's here, who's also the DVC Strategy, Acting DVC Strategy, Global and Corporate Affairs, who's with us, also from the Military Science Camp. And then Professor Nicholas Smith, the Acting CEO and Dean of Law, maybe in absentia. And then Dr. Ronel Retif, our Registrar. And then we've got Professor Elmi Muller, the Dean of the Faculty of Medicine and Health Sciences, who's with us today. Professor Peter Fontville, also the Acting Dean of Economic and Management Sciences, who's present with us today. Professor Vikas Fanikek, the Dean of Engineering, who's with us today, and part of the procession as well. And then Professor Ingrid Rewiski, the Vice Dean Teaching and Learning for Science, who's present with us. Professor Friki Scotts, the Vice Dean Research, Postgraduate Studies and Internationalization in the Faculty of Science. Professor Alf Porter, the Head of Department of Microbiology, who's present with us today as well. And then Professor Reginald, the Dean of Theology. Professor Kanshu Rajaratnam, the Director of the School for Data Science and Computational Thinking. And then I have apologies from Professor Guy Midgley, the Director of the School for Climate Studies. I did see the Dean of Arts and Social Sciences, Professor Fasterut, who's also present with us, but not really part of the procession. So I just want to acknowledge her, of course. I know she meant well when she came. So colleagues, today is really a special day, as we know it. It's an inaugural lecture, which is a formal event for newly appointed professors or those who have been promoted to full professor, which offers, of course, an engagement opportunity for the professor to share their knowledge, their scientific discoveries, scholarly journey, mentorship of students and early career researchers, as well as advancing their respective fields with the public and academic community. Similarly, it gives us an opportunity as the university community and all our partners and stakeholders to reflect on the journey and contributions and vision of the scholar who has advanced their respective fields and contributed to the frontiers of knowledge. So this evening, we have the distinct honor of hearing from Professor Fielding on the timely and thought-provoking topic, from pandemic to pandemic and everything in between. It is a title that hints at the depth and breadth of this scientific inquiry spanning virology, molecular biology, and the broader landscape of human health, as well as his commitment to advancing science that directly saves society. So according to cyber.com, which is a tool, which is a Scopus database, for the period 2019 to 2024, Professor Fielding's publications contributed to medicine, and I think the largest in medicine, about 71.4% of the outputs, which contributed to that. And I think Professor Elmi Muller will be very happy to know that the Dean of Science is contributing to health sciences. And then immunology and microbiology, and then pharmacology, biochemistry, genetics, and molecular biology, amongst other scientific branches where he's made contributions. His network involves both national and international collaborations. In addition to these contributions, the article with the highest field-weighted citation index is the co-authored article on coronavirus envelope protein, current knowledge, which was published in 2019 and has a field-weighted citation of 41.24, which is much higher than the university field citation impact. So that shows, I think, the value of that article and its impact. Throughout his career, Professor Fielding has embodied the values that we hold dear at Stellenbosch University. That's scientific excellence, collaboration across disciplines, and a deep sense of social responsibility. His work on human coronavirus, his leadership, of course, during COVID-19, and his tireless efforts to ensure that science remains at the heart of the public health conversations have earned him not only national but global respect. As we gather tonight, we celebrate not only his personal achievements, but also the Faculty of Sciences and also Health Sciences and Stellenbosch University's enduring contribution to solving some of the most pressing challenges of our time, from pandemics to planetary health and indeed everything in between. So without further ado, it is my great pleasure to invite Professor Fielding to deliver his inaugural lecture, and thank you all for joining us today. Professor Fielding.
Good evening. Sibu, thank you for that introduction. It warms my heart when I look out across the audience. The past month or so, we've lost some dear friends and colleagues that was with me at UWC. And when you're at those type of events and you hear all the good things that people say about the deceased, gives so much more meaning to this type of event where we celebrate people while they're still alive. So I appreciate you for being here. Thank you for taking the time. Professor Reginald was very, very impressed with this title. And he says for a scientist to come up with such a deep title is really impressive. And I think I made a mistake having such a deep title. Because as I was doing the slides, I realized, oh my word, so what do I do now? What do I present, what do I do not present? I think it's easier when you do your inaugural lecture shortly a year after you become a professor. Because then you can pick and choose what you're going to present. However, if you were first appointed professor, was it 13 years ago, and all of a sudden you have to do one at a different university, makes it very difficult. So tonight, I'm going to try to just touch on some highlights of the career, of my career, and when I say my career, it is, you'll see, it is not my work that we're celebrating tonight. It's predominantly the work of many of the people in this audience. Thank you to all of my ex and current students for being here, and many of my collaborators. I appreciate you. So as I said, I was going to make this a very deep, meaningful, high science presentation, and then I realized that the Dean of Medicine is here as well, so I need to keep it superficial, otherwise she'll be lost. So Almi, it's a pleasure. So this is in recognition of you. Just a career overview, very, very briefly. So I did my PhD, I graduated with my PhD 1st of January 2001. We really fell into the work on SARS. And when we get to the next slide, I'm going to just briefly tell you how I fell into SARS and the SARS field and the SARS work. You'll see Kraalbos over there as well in 2011. And this one I guessed. I guessed my work on Kraalbos started slightly earlier as well. But this is when students really joined the group working on Kraalbos. This was also the year where I was appointed as a full professor at the University of the Western Cape. And what makes it even more complicated, the academic standing in front of you tonight is the academic who he is, not only because of research, but is really multifaceted. And that's what I try to show over here. So I would have loved to have 2 hours to speak about all of this, but I don't. I have 40 minutes because I would still like to have a question and answer session as well. So you'll see in 2014 is when I really fell into leadership, and I the then Deputy Vice Chancellor of Research and Innovation at UWC, Professor Ramesh Barutram, coming to my office and saying, I've watched you in meetings, I want you to take over as the Dean of Research. And he left my office and I walked out of my office, and some of the students who were here were with me back then as well, and they looked at me and they said, oh my word, Prof, what just happened in the office? Your face is totally white. It's because ek het geskrik. I did not think of it. I contacted my wife Portia and I said, you will not believe what happened. But that same day I told Professor Barutram that I'm interested and that this is how my journey started in leadership. You'll see over there, there's a branch for coaching as well. So I did an MPhil in coaching at University of Stellenbosch Business School. And for me, this was important because as I moved into leadership and having gone through the journey that I've gone through, I realized there's one thing missing in higher education: support for academics, support for support staff. So that was the intent behind the coaching qualification. How do I support staff, students better? How do I equip myself? Was a bit selfish because that was lots of self-development in there as well, but I think that was just one of the extra pluses. And then the pandemic happened. And you'll see there's an offshoot science advocacy, and I was kind of forced into that as well by Gesanta Bader and his colleagues asking me, as the expert, are you willing to go out there and to speak about this? I said no. And then I started seeing the information going around. And I capitulated and I did my first interview on SAFM. They contacted me after 1 hour interview, live call-in show. never done it before. And they called me afterwards and they said, how did it go? Will you do this again? I said, never, never, ever again. 2 days later, I was on national television, saying something that was totally wrong, but that's how I learned. So as I said, I will not be able to touch on all of these. Very, very briefly, I have 38 slides. I have 40 minutes, so some of these are going to be very, very brief. The PhD. So my PhD was looking at a virus that was isolated from insects, from caterpillars collected in the Eastern Cape. And very interestingly, the farmers would collect these dead insects, mash them up. Don't know how much you can mash up that goo over there. Mix it with water and then spray it on the crops. And as soon as the caterpillars, and this is the one that I worked on, the moth that I worked on was the cabbage looper. It's a huge agricultural impact. As soon as the insects would eat, a day later, they would stop eating, they would move to the top, so they would move towards sunlight. They would become immobile, they would die and they would explode, and they would form that little goo mess over there. My supervisor at this point in time was Professor Sean Davison. And I don't know whether I can still call myself a molecular biologist after this project. So I ended up with 5 manuscripts after the PhD, but I realized thinking about this last week, what is a molecular biologist? And that's what I claim to be. I'm a molecular biologist. For each of the genes, for each of the portions of the genetic material that we sequence, when you determine the order of the genetic sequence, for each of those short pieces, took anywhere between 2 and 3 months. During the pandemic, researchers sequenced the entire virus within 24 hours. That shows you where we came from and where we currently are. So in the end, using very archaic methods, I managed to really assemble the genome of this 160,000 base pair organism, and that was my PhD. However, I became bored. So 2 years after starting, having 10,000 Rand from the NRF for my research per year, I could buy a set of Gilson pipettes and I have no money left, and I became really, really bored. So one day my wife and I sit at the airport, we're seeing off somebody, and I look at her and I say, end of next year we're going to be out of the country and I'm going to do a research fellowship somewhere. And she was like, this guy says a lot of things, probably won't happen. End of that year, we were on a plane, 2003, we were on the plane on the way to Singapore, where I was going to work on hepatitis C. And I was really excited to get to one of the top labs in the world and work on hepatitis C, something that has a global impact. So my wife goes with me, 3.5, 4-year-old child, and she's 8 months pregnant. We're on the plane. A month later, we meet with the government officials, Singapore government officials, saying, there's this new virus that's killing people in Asia. Stop everything that you are doing and work on this new thing. We know what the genetic makeup is. We have no idea what this does. Study this. This is how I fell into the coronavirus field, all by chance. So working 13, 14, 15-hour days with a very happy wife at home, with a newborn, a 3-year-old, and a pandemic lockdown, I still managed, and I still survived. And much of what I say tonight is hopefully also, welcome Mr. Rigo. Hopefully it will be an inspiration to those PhDs who are thinking, oh my word, I'm going through this alone. All of us suffer. Anybody who's done a PhD without crying at least 4 times during the PhD is not doing a PhD, they're lying to you. So very, very briefly, everybody has seen this picture since the pandemic. This thing is called the coronavirus because corona is the Latin for halo. And if you look at that, there's a halo around this virus. So the deadly three, so we have 7 human coronaviruses that we know of. 3 of them are deadly. SARS, the one that I worked on in 2003, MERS, Middle Eastern Respiratory Syndrome coronavirus that was identified in 2012 in the Middle East, as the name says, and SARS-CoV-2 that all of us know. So one of the things that irked me the most during the pandemic when people would speak about the coronavirus, it was one of the coronavirus, but it was not the coronavirus. SARS very quickly disappeared. Within 6 months, it totally disappeared. MERS is still around since 2012, still around. Only 3,000 people have been infected thus far. The bad news is that it has a mortality rate, a death rate of 37%. The good news is that it spreads very, very difficult between people. So typically it's spread between the camel and the camel farmer, or the person, the primary healthcare giver of the person who was severely ill. So imagine MERS jumps, then we're in for huge trouble. What is the common with the 3 deadly ones? The bat. All of these come from bats. Bats have adapted, and bats do not have any symptoms. But they can infect other animals, and from those other animals it gets to people. This is a figure I've taken from this paper over here, and you can see the pangolin, the dromedaric camel, and then a civet cat. See, these are the intermediary hosts that somehow becomes infected from the bat, and those are the 3 that spread it to people. What is the commonality between those 3? All 3 of them are food sources. So they're actually consumed in Asia, in Africa, and in other parts of the world. This one really irritated me in this figure because even though it says civet cat, it is not a cat. That is what it looks like. That's the palm civet. This is sold live in live food markets. This is where the original SARS came from. Who has heard of civet before? Who has heard of the palm civet before? 1. Have you ever heard of the most expensive cup of coffee in the world? Up to, what is that? Up to $50 per cup of coffee. Where do those coffee beans come from? The green coffee beans are fed to the civet cat, who eats those beans, poops out the beans. The beans have now been fermented. It is then purified and made into this very expensive coffee. So your fancy coffee that you fly to Asia for, it comes from poop. I'm going to preempt, since this is a Q&A session as well, I'm going to preempt. People ask me in each and every engagement, was this an unexpected pandemic? And the answer is no. This was not an unexpected pandemic. Very early on, I wrote an article with Mr. Morgan Morris where we spoke about the coronavirus family, and then a subsequent paper we spoke about, and this is really speculation. We speak about a possibly 4th deadly coronavirus that somehow evolved over 100 years to be one of the circulating ones now, and it only causes mild cold-like symptoms. So what happened? Evolution. So this virus evolved to become less deadly. So each time I was asked, are you concerned about this one and about the mutations? I would say no. From a vaccine perspective, yes, but from a evolutionary perspective and this virus becoming deadlier, looking at the other coronaviruses, the other 6, and the other 20, 30 animal coronaviruses, they would mutate, they would spread easier, but they will become less deadly. That's exactly what we're seeing now. Before, this was not the big pandemic, by the way. The big one is still coming. But that's a different conversation. Everybody has seen this as well. This is what the coronavirus looks like. Very, very, very simple organism. One of my favorite questions to my 2nd and 3rd years were, I made a statement for 10 marks. I would say, a virus is alive. True or false, and substantiate. And I would give them 10 marks potentially for true, and I would give them 10 marks potentially for false. because that debate is still ongoing. So you can see, very, very simple. I think by now everybody has heard of the spike protein, and to a lesser extent of these others. This is the genomic organization. So what are genes? What is your genome? Your genome is the part that determines your skin color, your hair color, determines who you are. If you look at the human genome, 3 million base pairs. The biggest of the 7, only 30,000 base pairs. So if compared to humans, very, very small. And yet, look at all the problems that it caused. Look at all of those. All of them, all of the coronaviruses, whether they are human coronaviruses or animal coronaviruses, they all share this makeup. Spike protein, envelope protein, matrix protein, and nucleocapsid protein, all of them have that. And then you have these what are called accessory proteins. And these accessory proteins are interspersed amongst these structural proteins. And just look at that. Very early on, 2003, we sat down and when we looked at these, we said, if you look at the number of these accessory proteins, why do the deadly coronaviruses from humans have more accessory proteins? Could it have a function? And at that point, researchers were not even sure whether these proteins do have functions or not. So those were very deep questions, questions that we had no answers for, and that would form part of our work. Even though they are named the same, these are not the same proteins. So SARS-3A is not the same as SARS-2-3A or 3A in the other coronaviruses. They are very, very unique to the particular coronavirus. So I say that the virus itself becomes the target. What do I mean by that? The more we understand the virus, the more we can treat, the more we can prevent. And hopefully we'll see more of that now. So if we target the virus specifically, the intent is either to stop the virus after it infected the cell, or to prevent the virus from entering the cell, or to prevent the virus from leaving the cell. So we can target the virus, but we can also target the human. But what is the biggest problem with targeting the human? If we target the human, how do we know that what we're targeting will have a specific effect and only affect the way the virus interacts with that cell? You could have a systemic effect as well, and then you're in trouble. So that one is a bit more challenging. So just some more background. This is a cell. So let's say this is a cell in your lungs. So you breathe in this virus, it moves, and then it attaches to a receptor, gets into the cell, it takes over the entire cell. So the cell stops making cellular components. The aim of a cell is to make components for other cells and then die. This virus gets into the cell and keeps the cell alive for longer periods of time. And it keeps the cell alive for longer periods of time because it's now converted the cell into a factory for viral components. So pretty soon you have unhealthy cells. You have cells that want to die, and the virus are keeping them alive. Once a virus leaves into the bloodstream, so more virus was made here, and then they leave into the bloodstream or out back into the respiratory tract, good, others can be infected. But that also exposes the virus to drugs and to your immune system. So what does this virus do? This virus has found ways to spread between cells without ever leaving the cell. And that has been shown to be a function of the spike protein. So all of a sudden, you look under a microscope at an infected person and you see these huge cells with no membrane between the cells, but multinuclei. So cells are fused. So the virus never leaves the cell. So it's never threatened by the immune system, it's never threatened by drugs. Are these things alive? Because they are certainly clever. They've evolved. There's also a phenomenon called unproductive infection. So a virus can get into a cell, but it never produces more virus. And that could be due to something wrong in the virus or something wrong or right in your cell. We're still trying to understand that. Why are some people more susceptible to severe infection? Why are some people susceptible to infection? Why are others not? We have some idea, but I don't think we're close to understanding this. So the real journey, I said, started in 2003. And very, very quickly, we took the genetic material of SARS, we chopped it up into bits, we made the proteins artificially in the lab, we injected these into rabbits, into mice, so we made antibodies, so we could study the virus in clinical samples, but we could also take the chopped up bits, make proteins artificially in the laboratory, and then test it against blood from humans. So very quickly we saw something very interesting. The nucleocapsid, the most abundant protein in infected people, that's the protein that interacts with the genetic material of the virus to keep the genetic material stable. So present in very, very high quantities. And then we saw a protein that nobody expected to even be a real protein. And initially we called it U274 because we were the ones identifying it. Uncharacterized protein of 270 amino acids, so the size, and the initial papers were accepted, and then the gurus in the field came back and said, no, there is a convention for naming these things. You will not do that anymore. So we had to change the name to protein 3A, which is part of the convention. So what made our detection kit different? So this was the world's first rapid 15-minute detection kit for SARS. And it was different from subsequent ones because we looked at the nucleocapsid protein in the infected person, and we looked at this accessory protein that nobody thought to look at. So you would take a drop of blood or sputum from somebody, put it in over there, and if you have a positive, you would see the 3 lines. Sometimes in some patients, in about 10% of patients, you did not see the bottom line, but this was the negative. 2 patents and an invention disclosure. New PhD graduate, and I'm thinking, whoa, I'm going to make tons of money. That's why we're all in this, tons of money. I don't know whether I should say for the better or for the worse, the virus totally disappeared from the human population. So isolation worked for this one. Remember what we did, what we tried to do during the pandemic? We tried to isolate. Didn't work. With this one, it did. So I made absolutely no money on this one, even though the kits were available because it was licensed out. And I always feel guilty for wanting to make money because people would have died. In the end, fortunately with this virus, only 8,200 people became infected in 28 countries, and 10% of people died. So whenever I would do this lecture, my students would look at me very strangely. So why would we be concerned about 800 people globally who die? And I would tell them the story of a family in Singapore where dad, mom, daughter, and son, 4 medical doctors. The mom was in a coma. And when she came out of a coma, they had to tell her that the entire family was infected and she lost the other 3. So it does affect somebody. It does impact somebody. And that's what we need to keep in mind as well. Apparently you need to press the right button. So as I said, was this unexpected? No. This was written in 2013. And this is, in particular, I wrote this for Fun Week, and it was about the global impact, financial impact of the coronavirus that caused the common cold, and it's billions that we're losing yearly. due to these viruses. But I also referred to MERS. And my biggest concern was, if MERS is located in Saudi Arabia, what happens yearly in Saudi Arabia? The Hajj, where millions of Muslims go to Saudi Arabia. Millions of Muslims go home. What happens if this virus remains pathogenic, so remains deadly, can cause disease, and it spreads easier? That's what this article was about. 2013. And I made that statement, saying that we really, really need to be prepared as a scientific community, but also as a government. Because global travel have become easier. We're now moving, exploding human population globally. So we're moving and we're in much closer contact with wildlife. These viruses are zoonotic. They come from animals. Huge problem. Lo and behold, the pandemic. So over 22 years, yes, we looked at spike, we looked at nucleocapsid, we looked at envelope, and then we looked at the accessory proteins. And these were the ones that were not sexy. These are the ones that nobody wanted to look at. And we said, but they're here for a reason. Why are they different? So we looked at those as well. So I mentioned that we looked at spike. Virus comes in, and it binds to a receptor, and then it moves into the cell and it takes over the cell. Who knows what that receptor is called? What is the name of that receptor on the cell? ACE2. ACE2. I'll come back to that. So we looked in that previous slide, some of the publications from the collaborative antiviral group in Singapore that I was part of, where we looked at the spike protein. Once again, chopping up the spike protein, a huge protein, chopped it up into various parts, and then studied each part to see which of those parts are the most important in the human body. Which of those are important for this virus getting into the cell? I think another favorite question that people normally ask me, so why do we have these receptors? Why do we have ACE2 receptor in our body? That's dumb to have a receptor in your body that a virus can use to get into your body and to make you ill. These are proteins with functions. Those with high blood pressure. Many of you use ACE inhibitors. ACE2 plays a role in controlling blood pressure. These viruses have evolved to use and abuse the ACE2 receptor to get into the cell. So see what happens. It attaches, and then those sites that we've identified, those are the sites that's most important. You have a conformational change in the protein, then it attaches to the cell membrane, and then it gets into the cell. If we know which of the regions are the most important, what does that mean? If we target those regions, we can prevent the virus attaching to the ACE2, or once it's attached, we can prevent it from being processed so that it's pulled to the cell surface. That's what people have been trying to do. So with vaccines, you have antibodies that either attaches to the virus, to the parts of the virus sticking out, the spike protein, and it cannot attach over here. But now imagine you can identify molecules that prevents that attachment as well. Imagine you can identify molecules or enzymes that prevents that processing over here. You would not have any of these. That's what our work on spike protein led to. However, we've also looked at spike for other reasons. Spike is very, very, very important for immunity, and that is what this is all about. We currently have a master's student registered at Stellenbosch that's also looking at the role of spike and how it evolves under immune pressure for MERS. So we're still ongoing with that. I am still concerned about that one. I don't think we've seen the end of it. Then I've mentioned the accessory proteins. The accessory proteins are those that are unique to a particular coronavirus different between the different coronaviruses that nobody thought had a function. Well, I can tell you now they have a function. Tasneem Suleiman was in Germany when she did a PhD with me. They assembled an entire virus artificially, knocked out some of these, and saw that they do have function. So we've done some very, very interesting work to show that they indeed do have function. What those functions are, still evolving and we're still learning, but we don't know yet. That's my favorite one over here, thinking I have finally made it. I might not have made money, but I have now named a protein, only to be told that you're not allowed to do that. So it's now no longer U122, it's now 7A. We've also shown that first one that's encircled, these virus accessory proteins also interact with the host cell machinery. If it interacts with the host cell machinery, what does it mean? Your cell cannot make more cell components. So the virus manipulates the cell into making viral components, which means that more viruses are made. That's what the function of one of these accessory proteins. So just to give you an example of how cool this is. So one of the projects that I had in Singapore was to look at one of these accessory proteins. So this is U122, that is now called 7A. Had the genetic material, had this into a system where I could artificially make this protein. And each time I made this protein, it was very, very weird, because I would see two forms of the protein. And the longer the cells were kept alive, one of the forms would disappear, and the other one would become the dominant one. And I could not understand this. So I made some mutations in the virus to show that once this protein in the cell is made in a particular compartment, the cell's machinery, enzymes, cleave that site right up there where the arrow is, and this protein moves somewhere where it's needed by the virus. This is basic research, nothing to do with drug development, nothing. You can see how broad the research field is. And this was so cool. I remember going to a conference, and I think probably the leader in the field, Professor Christian Drosten, and one of his PhD students came to my poster and said, "Have you submitted this for publication? Because I think you've just," and it was not cool for killing the PhD thesis, but to show that we're doing impactful work. And the PhD student said, "I think you just killed my one chapter." And he asked me, explain why you seeing this, we're seeing it in the patient now, but he could never see it in his artificial system. And to the students in the audience, this is where I learned the phrase, in our system, this does A, B, C, D, E. So I was very fortunate in using the particular type of cells in the lab, using the particular plasmids to express this, and by chance I saw this, and then I could study it further. So, in our system. Keep that for your papers, please. So, cool. I said this protein is made somewhere, and then we have the enzymes of the cell cleaving this, and then it moves somewhere else. And that's what it looks like. And once it reaches its site, it can now be incorporated into the virus, and you have a functional virus that's released or moved between cells and can then infect other cells. And then probably one of our most impactful research. Sat at home and I thought, we had lots of time during the pandemic. Remember, we couldn't go anywhere. So this was one of those where I thought, if all coronaviruses have envelope proteins, and we now know that envelope proteins play a role in how deadly the virus is. Why? All of them have envelope. Envelope determines how deadly it is. There must be differences. So I spoke to a PhD student, Devald Skuman, and I said, can you look at these proteins? Try to figure out the reason. This was the review we wrote. The review was published, I think, 3 months before the pandemic. To date, it's been cited 2,700 times. All of a sudden, everybody's looking at this protein. All of a sudden, this has changed the field in that particular area. His subsequent review said, so everything that we've looked at, how does this determine that how does it contribute to how deadly the virus is? Which led to that modeling paper. Hey, where did my other... And if you compare these envelope proteins, look at the 3 deadly ones. These are the 2 non-deadly coronaviruses. We studied 5 of them. These are the deadly ones. And that small little portion at the end of the protein, in the deadly coronaviruses, it has a little tail, and this tail can move. And because the tail is flexible, it appears as though that flexibility allows this protein to interact with human proteins. And because it interacts with human proteins, it stops those proteins from doing what they're meant to be doing, which means that cell becomes dysfunctional. Is this what it could be? Is Devil in the audience? No, so I can say his examiner report just came back. And the examiners were saying this is the first time that this has been shown. By the way, at UWC, you're not allowed to tell students what the examiners say until Senate Heidegger has sat. They have not sat. So he's not in the audience, I've just told you. So apparently this is very, very significant. This paper has just come back from the reviewers, some minor changes, so hopefully it will be published. Is my HOD in the audience? You asked me before this were my publications. This is the first one that will be published probably in the next 2 months. You love that. That's all he's all about the publications, I'm sorry. So this one, I think very, very significant because as the reviewers said, this is the first. The reviews of this paper said the same as what the examiner said. This is the first time that this has been speculated. And hopefully by end of July, with Professor Samuel Igeye from the School of Pharmacy at UWC, this other paper will come out. So we've screened small molecules that could potentially be interacting with that little tail. And we've identified certain compounds that could be effective against the deadly coronaviruses. Huge potential. We could then essentially manage and control the current ones and any future ones that we don't know about yet, if the tail is active. So I think this is quite exciting for us. So our potential targets that we've identified, all of those little red, what do you call those? 8 blockers. Those little red circles with the little cross through it, those are all the areas that some of the work that we've done could be potential targets against these deadly coronaviruses. So we're studying some of those. How much time do I have left? But anyway, I'm nearly done. During the pandemic, we had lots of time. So I had lots of time to think. And one of the questions I was asked most frequently during my interviews was, why is Africa not hit as hard as the rest of the world? And we said the young population, and we said it could be genetics, and we said it's the weather. Nobody spoke about diet. So once again, I contacted 5 or 6 colleagues, and I said, I have this idea. Could it not be something to do with obesity and starvation and diet? Nobody wanted to work on this opinion piece. Once again, I spoke to one of the students who said, of course he'll work with it, it's a publication for him. And very excitedly, 2 months into it, he said, I think this is a plausible opinion. And I've just read a few papers that speaks about leptin and decrease in leptin levels in other virus infections and how it impacts on infectivity and how deadly something is, a viral infection. So eventually we started at the journal Lancet. So we aimed really, really high. And 6 journals later, it was finally accepted. And each one of the previous 5 said, you cannot say this. Even though it's your opinion, there is no scientific evidence for this. It's an opinion piece, substantiated. Eventually it was published. Since this paper, groups have looked at leptin and diet and the impact of it. So our hypothesis in conclusion of the year was weight loss or starvation, low amounts of leptin, so you have an increased risk of infection, but a reduced risk of hyperinflammation. What was one of those things that each person that became nearly each person that became severely ill and very often died had in common? Hyperinflammation. That's why obesity was a very high risk, hyperinflammation. So I think this was a piece that I still wish it could go into Lancet. That would have been so cool. Very briefly, I'm going to rush over these. I do not have the time. Kraalbos, 2018, as the molecular biologist, the person in the picture, Professor Jeremy Claassen, asked me to be the molecular biologist on a project. I'm trying to understand why this plant can kill fungi and bacteria in an agricultural setting. And we were looking at this, and then about 3-4 weeks into this, I said, Jeremy, and by the way, you know how this started? Jeremy remembers speaking to his in-laws who are from the Northern Cape, speaking about this plant, speaking about how the Khoisan people used it on the skin as a lotion, how they used it for eye infections and various diseases. And he thought, why not test this plant? So 2 weeks after joining this project, I said, Jeremy, has anybody looked at the cancer properties, anti-cancer properties of this plant? He says, no, try it. This was teaser in uni, and teaser eventually showed that it does have anti-cancer properties. There are 2 students that here, either in person here tonight or online, that's looking at this plant. We have now managed to break this plant down into the components, and we've identified the components that has the anti-cancer properties. We've now combined those, and Asia, you can tell me if I'm lying or not. Asia Chafikar is the PhD student working on this. Combined it with nanoparticles to target. These things are very, very specific. What did the indigenous people use it for? Skin ailments. There's some knowledge in this boererate. They work. We just don't understand why. Unfortunately, this was not our paper. This was the paper from UCT, one of our collaborators. They published as well, but I guarantee you, we were the first group to look at the anti-cancer properties of this plant. Dean Solomons has also now shown that this plant has potential to treat Alzheimer's. He's tested this in an Alzheimer's model, some very, very promising results. ASIA received TIA funding to test the anti-cancer properties in a mouse model. And then I came to Stellenbosch, and we had to stop the research. So we had to give the money back. Don't worry, Professor Moya, we're already talking to Innovis. We'll bring it here eventually. Science advocacy. Why did I agree to go on radio, to do what goes against the grain of every scientist, every academic? We are not trained to do this. The WHO very early on said, we will not only have a pandemic, we'll also have an infodemic. Everybody became an expert. Because of the internet, everybody shared information. 98% of that information was incorrect, but everybody was an expert. So it was really, really my wish to go and to engage with as many media outlets as possible, to share my knowledge as an expert. And I learned very quickly. You say, based on the current information we see. My first interview on television, remember I mentioned 2 weeks later, I went and they did all the makeup, and I'm sitting under these bright lights, and she says, so Professor Fielding, should we be worried about this virus? And I said, if we look at SARS 2003, Don't worry, this will disappear. I think I was slightly wrong. 4 weeks later, same studio, I was asked the same question. I said, you know what, based on what we know now, this is probably going to become a circulating virus. So I learned very, very quickly, we need to explain to the general public that what we say as scientists is based on what we currently know, but that can change. So, Professor Moya, this one was after I joined Stellenbosch, speaking about influenza. So we can do some very informative, very broad presentations. This one was really, really cool. It was just after Donald Trump came back to the White House, after he contracted SARS, after he contracted SARS-CoV-2, so COVID. And remember, he said he did not say that you should drink bleach. That was that was fake news. Remember, you got this whole cocktail of everybody wanted to know if that is true. That's what this interview was about. So very topical, and it was shared elsewhere. And then every so often we do things where we look like a clown as well. And I was trying to figure out what that one was. This was actually an interview where I was asked about, and the entire 40 minutes was about the Chinese now trying to use anal swabs instead of nose swabs for the testing. I think that's why I look like that. So how do we communicate? I think all of these are important. Science advocacy is about taking out our message and explaining it and sharing it in a way that everybody can understand. And it will be picked up. And just for the record, that vaccine article I wrote, I am not an anti-vaxxer. I've said from day one, when one day for this disease we have vaccines, we should target those that are high risk. That's what this should have been about. Should not have vaccinated everybody if you look at classical infectious disease history. So very often I was clubbed by the anti-vaxxers and I was clubbed by the vaccine alls because I was somewhere in the middle. So if you're going to do science advocacy, you're going to be clubbed, I promise you. You can never, never make everybody happy. So I'm drawing to a close. So what is all of this about? How do I measure academic success? Is it about these products? Is it about the papers? Is it about the millions that I did not make with this product? I don't think so. For me, it's about that. It's about having students that can go out and that can do these opinion pieces. It's about the students going and doing the interviews. There's Neem going out and speaking on so many radio and television shows. Why? So Tasneem did a PhD with me. She joined another lab as a postdoc. She worked with collaborators at Stellenbosch. They were the first ones in South Africa to isolate this virus. So when I was asked in interviews subsequently, is this real or is it caused by 4G? I could say, one of my ex-students isolated this virus. That's what this should be about, the lasting impact. These are going and being a role model for other female scientists. And she was interviewed for that. Dewald going, not only to schools, but doing an interview for Chinese television. And do you know how cool that look? When they dub you and you speak in Cantonese during the interview. It's the coolest thing. For me, that's what this is about. Making an impact through our students. So finally, my acknowledgements, a whole list. If I've omitted anybody, I am so sorry. I apologize. Those three at the bottom I've highlighted because these are the three postdocs. One of those we share with the School of Pharmacy at UWC. The other two are here. Looking at long COVID and looking at nanotechnology for treating TB nogal. For treating TB. I'm a molecular biologist, so it's wider. These are the students. The work that you see is due to these people. They do the hard work. They're the ones who stand in my office and they cry. And then they'll turn away from me, stand in the corner and cry and look back at me and say, okay, Prof, we can talk again. Because it's frustrating and yet rewarding. And I forgot to add the collaborative antiviral group, where I did my 3 years postdoc, my research fellowship. Learned a lot, suffered a lot, and you'll see, and you're probably thinking, oh my word, he's not acknowledged his wife and his sons. Because they shouldn't be on there. They're sitting here. So to my family, to Portia, Daniel, Michael, and Ezra online, thank you. Thank you for being understanding. I know I've not always been there. Difficult times. I think when you're a researcher, you can be swallowed up by research very often. But thank you. Thank you for your support. To have a wife that will get onto a plane, 8 months pregnant, with a 3-year-old. Need I say more? No. Colleagues, thank you. Thank you so much. Oh, questions? I think we have time for 2 questions, if there are any questions in the audience, please. Not everybody at once. Ach, nee man.
So do you not feel nervous that we've stopped vaccinating? And do you not think we should continue to vaccinate because we've got them now and we know another pandemic is coming? So what would be your advice?
So I've never been concerned about the SARS-CoV-2, the COVID-19 pandemic. And listen, all of us have lost somebody very close to us, so I'm not minimizing the impact of it. The WHO has said the next pandemic will be caused by pathogen X, will cause disease X, and in 2003 the WHO was saying 200 million deaths globally. That's what the next pandemic is. So I think with this COVID, we need to be treating it, and now I'm going to sound like Trump, so excuse me. We should be treating this the same way we're treating influenza, seasonal influenza. We have these now, we now can make fresh vaccines against the predominant strains, so we should be targeting those who are susceptible to severe disease. So those with multiple comorbidities, the obese, etc., etc. If we target those, I think we're fine. If we're in contact with somebody who cannot have the vaccine because of whichever medical conditions, and they're at risk, then those in their family immediately should take the vaccines. So I think that is the wise way of doing this vaccination program. Does that answer your question? Yeah. Samuel.
Yeah, thank you very much. Mine is from the perspective with advocacy, so-called ambitious. Now 60% of our vaccines made in Africa. Do you think the research can feed into that climate change to bring about the realization of that dream? That's one thing.
You mean in general, the research we're doing in South Africa in general?
Are we feeding into that in order to enable that realization? Not only for the disease X, but even for those regular yearly infections that we have.
Samuel, and I mean, I know that you work very closely with the CDC and WHO, etc., United Nations. The problem with Africa is not the human capacity. We have the most brilliant researchers in the world in Africa. The problem, I think, are the resources, the funding. How can we, as a country, when one person decides to cut funding, panic as a country? How have we become dependent on our sustainability on other countries? And we're going to replace one master now with another master. So we're going to find another country to invest. So I think if we do things wisely and we invest where the money is or should be, I think it's probably still a bit ambitious, but we could probably fill a huge void. So I'm a bit concerned about the wastage of the resources. But you know, I mean, the most brilliant people from Africa, and that's why we are so popular when we go abroad. Colleagues, those are the 2 questions. Thank you very much for your time and your attention. I appreciate it. Thank you.
Well, thank you very much, Professor Fielding, for that absolutely fantastic lecture. But I think you all will agree with me that even more impressive, as you walked into this venue, was the young Sheldon pose by Professor Fielding in that title slide.
Thank you. I did not choose that picture, by the way.
Well, good evening to the Fielding family and friends, to the leadership of the Faculty of Science, to our university leadership, colleagues, ladies and gentlemen. It is an absolute privilege and pleasure to be with you tonight and to be part of and share in this inaugural lecture, which is effectively the introduction of Professor Fielding's academic scholarship and his research interests to the academic community at Stellenbosch University, but also to our community at large in the town. Now, as has been mentioned, the inaugural lecture is typically a rite of passage event within the university and marks the recognition of a colleague that has been promoted or appointed and entered the ranks of full professor. In the case of Professor Fielding, it is the recognition of him bestowed the rank of full professor on his appointment as the Dean of the Faculty of Science. You were sharing with me outside that this is actually his first inaugural lecture as a professor. I think many of us that have joined this university have had more than one inaugural lecture, but this is his very first one. So it makes it extra special, not just for him, but also for us as an institution. Now, this event affords the opportunity to Professor Fielding to have shared with us his innovative ideas, his discoveries, his deep insights with regard to his discipline focus and his academic scholarship and interest. In the case of the lecture tonight, Professor Fielding has shared the insights on pandemics, and in particular the coronavirus. Now, thank you, Professor Fielding, for your lecture. I have to say, when I read the title of the lecture for the first time, I mistakenly thought it was From Pandemic to Pandemonia. because that's what it felt like in 2019, 2020. So thank you very much for the lecture, From Pandemic to Pandemic and everything in between. Thank you for that deeply engaging and thought-provoking lecture. You've taken us on a journey not just through the timelines of global health crisis, but through the very fabric of how society responds, adapts, and perhaps most importantly, remembers these events. You also shared with us your journey as an academic, and also in terms of your personal development, encompassing coaching, advocacy, and leadership. So the title alone, From Pandemic to Pandemic and everything in between, hints at a vast, complex terrain, and you navigated that with both intellectual rigor and a remarkable sense of humanity. You reminded us that pandemics are not just medical events, they are social, political, economic, and emotional turning points. They expose our vulnerabilities, but also illuminate our resilience. What resonated most was the idea that the in-between, although you didn't really get into that too much, is just as critical as the crises themselves. It is in these intervals, these quieter, often overlooked chapters, that systems are rebuilt, memories fade or solidify, and the seeds of future preparedness or forgetfulness are sown. Your insights challenge us not to wait for the next global emergency to ask the hard questions or take meaningful action. Instead, you invite us to see continuity, where there's often a temptation to see rupture, to view public health not in isolated spikes of urgency, but as a sustained commitment to equity, truth, and care. Now, one of the indicators of a good inaugural lecture is the ability of the professor to share deep and complex disciplinary knowledge in a manner that is both understandable and thought-provoking to the general audience, but also clearly articulates the novelty and the complexity of the work to disciplinary experts. This is an incredible balancing act, and you have done impeccably well in that regard today. I mean, I as a novice, started to understand the coronavirus. This is the art of a true professor. Congratulations once again, Professor Fielding, on your body of research and work on the coronavirus and public engagement in bridging the gap between scientific research and societal impact. Your work does not simply add to academic knowledge, but truly enhances our understanding and our insight into researchers' evolving responsibilities to advance scientific knowledge and address global health challenges. We are extremely proud of you as an institution for your work as well as for your achievements. Keep up the fantastic work in helping us understand and improve our response to global health challenges. In commemoration of the inaugural lecture, it is my great pleasure to hand over the scroll to you.
Good evening, ladies and gentlemen. It's my privilege to say a few words of thanks. Professor Fielding, as a microbiologist, I totally enjoyed your talk, really. I must just give you a little secret. The first words from my mouth when I first met him as his HOD were, "Where's our articles? Where's the scientific articles?" Remember? Baie ongeskik gewees. Anyway, he did not disappoint. As you can see here, it's flowing in. You are a role model for many of us. Thank you. Okay. But now I have to really thank the most important people in the room. Portia, where are you? Where are you? Thank you. Thank you for supporting this guy during the years. I know that he's sitting there in his speech here. It's because of you. Okay, sorry, Bertie. And also thank you for, there they are. Yeah, Michael and Daniel, you are here. Thank you for your patience with your dad. All right. Thank you. I know, being an academic myself, it's sometimes very horrible for the for the kids at home. Okay. Yeah. For the students as well. So, yeah, I just want to also, online, Ezra is online, eh? On the other side of the globe, busy with his MSc, I think. Was it MSc? PhD. Oh, excuse me. All the best, eh? It's, where you are, if you can see me. Yeah, there. Yeah. He's there on the other side of the world. Yeah. Just don't cry too much. So, yes, then I also want to thank all Prof. Fielding's friends and colleagues from UWC. Unfortunately, we have him now. And his colleagues from Kraalbos who attended this lecture with us here. I forgot. Anthony Scholes? Thank you for supporting him. I forgot about you. Sorry, I just recognized you in the. Thank you for being here as part of the family. Okay, then I also have to thank the team that made this possible, this event. And this is, I don't know if Jonathan Dale was involved, but I know Amira Brown was involved. She actually ran to the office. Thank you for bringing my vleermuispakkie. Okay. And Krista Davids, thank you so much for your input. And Kuma, where is she? Is she out there? Tell her thank you. Yes, and Suzette Els from the faculty. She's here. Thank you, Suzette. I know you didn't want thanks, but yeah, you were my, how can I say, my information node. We got most of the information. Yes, and then Prof. Derish, Prof. Moyo, and the rest of this. I can't name you all. You're too many. You're not so important. Thank you. Thank you for taking your time here with us. And so, yeah. And now, guys, I have got the pleasure to invite you for refreshments just outside there. Okay, that's been prepared by Stias, and the food is always good, trust me. Thank you guys.